62. Maybe I was 20, and Professor Sitaram Iyengar 64, 64, somewhere around about it. And HR Ramakrishna unfortunately is not attending it because he had got some problem of his health. 3-4 people were there. Gopinath Kuddara, he was also there. We have that. 4-5 people, with the sole object of popularizing science and to bring the latest developments in different branches of science and technology to the common audience. Maybe on that day, we never had such a room and other things. We used to sit in a small room with some Broadway chairs like that, making noise also, 4 or 5 chairs, 10 chairs like that. And Professor Nasimaya was a teacher. We were students. Age difference is 25 years for me and Nasimaya. And when we used to attend things, people were making noise or fun or something like they used to say that, how can the science form go? And they're all cheer us. Nasimaya forced them to sit, otherwise nobody will come, something like that. But imagine, compared to that day, what sort of a hall we are having, and instead of 4 people, I think almost the hall is full today. That in Nasima's stand is vindicated completely. And started on Wednesday at 6 o'clock. Naturally, some people used to come, some people may not come. Nasimaya was also worried after 5-6 years, the people are not attending, the strength is not there. Suddenly one teacher suggested him, sir, I should not do it on Wednesday, do it on Thursday. There is no logic here. He said Thursday. And Nasimaya accepted it. The problem was, the person who suggested never turned up. Like that, some other person suggested, sir, 6 o'clock is bad time. You should do it 4:30. We also accepted that one. We started at 4:30. But the person who gave the session never turned up. That we lost one more person there. Therefore, the clear-cut objective of popular science, only Wednesday, just imagine the whole world, either any what's called comparable situation, started in 1962, today it is 2016, 54 years, we have not let off a single Wednesday. Not a single Wednesday. Of course, Kashyap was not lucky because he had left us to Harvard University, But Kashyap is at Australia, one Dr. Kashyap is sitting here, highly enlightened person, and coming from Harvard University, Purdue University like that, but has created one record. If any of you were to break it, I will give you 1 crore rupees now. That is, he was the first person to get first rank for National College of Assembly. Second rank is the first rank to Pretam Rogo. Athwa, neither first person, but first person even if you get first rank, you become the second person. Impossible to break that record, no? That is Dr. Kashyap. In fact, I am very happy that he is sitting there when a young person, if I am correct, he should be somewhere 28, if I am correct. I think from this platform, the youngest person to deliver a lecture, and he is sitting here, that is Dr. Vijeth Iyengar. Shortly, in few minutes, he is going to deliver the talk, and his talk happens to be 2713 number. And Yatik number when Ramanujan was in the hospital, somebody said, some taxi number, suddenly arranged it with some cubes or squares, something like that. I think my lecture also somebody may do on some day, 2713, the lecture, and shortly he is going to deliver the one. He is from the Duke University. The name itself suggests it is the 8th number university in America, and Institute of Neuroscience and Psychology. Long back, people used to respect physics as a grammar, nothing more than that, But no, nobody is coming to physics, chemistry like that. Neuroscience means everybody says, hello, we pay 250 crores, 300 crores, 400 crores like that. It's a very rich department, neuroscience. And he's going to talk on how memory is stored in the human brain. Today, on behalf of the institution, on behalf of you, on behalf of me, I extend a very cordial welcome to Dr. Vijeth Iyengar. I speak until, I think for 5-6 seats on the video. That is head of the country. But it will go over, still has some more points are there. Sitaram knows about it. See, my welcome speech depends upon the occupancy. If it occupied half, I will go on until it's filled up. And education, PhD, Duke University said, psychology and cognitive neuroscience. I never used the word cognitive because it needs some specialization. I am using only the neuroscience. MA from Duke University, BS, Tulane University, Cell and Molecular Biology. Cell is important, molecular biology is important, and Christian biology is more important. We had a subject. Then they got lot of awards. If anybody gets a better award, already the seats of they're going around like this. Afterwards they can come here. If anybody is there who has a better award, they can stand up. 2016 US Presidential Management Fellow Award. Just imagine the top. Very competitive award, selected among 1 lakh applicants. Oil and only 1 lakh. Just imagine. Only 1 lakh. 2010 to 13 National Science Foundation Graduate Scholarship. Well, that was obtained by Satish Chandra. I think he's going to speak elsewhere. Same scholarship in it. And 2016 Duke Alumni Association Forever Duke Award. Then 2007 National Institutes of Health Exceptional Semester Student. Internships and research experience. US National Institutes of Health. Then our Professor Sienna Ross Place. Then Louisiana Department of Health and Hospitals. George Mason University. Publications co-authored or authored 5 peer-reviewed papers and approximately 18 investigation national conferences. Of course, you may know already, he is the distinguished son of Dr. Sitaram Iyengar, who is sitting here, third son. Of course, naturally, he has to be first son. I don't know his age at that point of time, when he gave that class at the size form talk. By that time itself, he had isolated some gene regarding cancer and other things. He is also working in a great place. And second son is also there. First, second, third. Sitaram Iyengar is so proud. He wrote a letter to all of us stating their achievements. I've seen that letter also. And apart from that, Sitaram Iyengar, whenever I invited him, whenever he comes here and delivers the lecture, of course, generally, if somebody delivers a lecture, you will get an impression he may get TA, D or something like that. But out of 2,700 lectures, we have never paid TA, DA to any person, even the local laureate. At best, we have paid him good phone value. Nothing else. Imagine 2,700 lectures, if I start paying TA, DA, it's impossible to conduct it. Even if you spend 2,000 rupees as a taxi charge, actually he has brought a taxi which he is taking, and therefore, it's impossible to conduct it, apart from the remuneration. In fact, Genome Hunting from Lipkin, future Nobel laureates from British Columbia, he gave here, we never paid him anything. Dr. Negation from Purdue came, never gave him anything, no remuneration like that. And in fact, that's why I appreciate Sitaram. Not once, if I'm correct, I saw him with his first son, long back, let us say. From that day onwards, maybe 20, 30 years, every year has come and given. Apart from that, the greatest thing for us, I think no world student has done it. He has granted us a transdisciplinary research center from Florida International University. We will make an inspection after 1.5 years. When they come after 1.5 years, we have to say we are not done any work, what should we do now? But suddenly Professor Mudra, she is sitting here, she comes with the paper, and when I gave it to Sitaram Iyengar, he saw it and tells me, it's a wonderful paper, it's of some standard, And then 3-4 days I can research start again. Already we got 2 students, some 3-4 papers. That's how this research center today. Professor Dineshan, he is such an important person, professor from International Institute of Technology. He is going to retire from that. I told him to come and join our center. Like that we have to build up, there's no way at all. I don't think in 100 years, no student has tried to give us a transdisciplinary research center, recognized one from a university which is at the 45th level in America. It's not from somewhere 2000 university or 3000 university like that. I think we have to give another clap to Dr. Sitaram. I think more school, therefore I will end my welcome speech. And then, tomorrow, we have got one important person, Dr. G. Padmanabhan, former Director in Institute of Science. He is called Father of Vaccine, GP. He is called a Father of Vaccine in Indian conditions. He is going to talk on translation efforts in health research. Or Janta or Ole person, Janta Ole scientist today. someone is scientist, they think that he may get a lot of salary or something like that. It tells from this platform, a scientist, I'll give my example, he says, I am still living, his age is 75 or so, I am still living in my father's house, I have not built a house. First condition. Second condition, none of my work gets me a Nobel Prize. In India, everybody thinks they will get a Nobel Prize. No? He stays on the platform. I was sitting in front, I was laughing, I told him, somebody wants Nobel Prize, nobody will give you. Somebody does not want Nobel Prize, if it deserves, they will keep it. But no one very clearly says, none of my work will deserve Nobel Prize. Same thing Balaram also says. In fact, we should be grateful to both the people because they are given every year 39 years, one talk or the other. Then on 18, Shana Olsen, she is from Yale University, Duke University, Carnegie University, several things are there. She is speaking on tracking the objects of insects' affections across species and continents. Then we have got Govind Rajan, Professor, Chairman, Supercomputer Education, speaking on supercomputing. The subject and think about supercomputing. Next is race for ranking or science. So we can put in some questions. No? Like that it will go. I think almost full. I will stop my welcome speech. I request Dr. Vijeth to continue with the lecture, sir.
Good evening. I'm especially delighted to be back here at the Bangalore National College Science Forum. Professor Ram Rao and I were remembering a memory back about 10, 15 years ago, wherein my father, who was in the front row here, was giving a lecture, and I was sitting there advancing the slides. So it's funny how time flies by. But before I begin, I want to give Dr. Ram Rao a hand in helping organize this. It's extremely humbling to be here, knowing that tomorrow's lecture is by the father of the vaccine, and in the coming days, head of the Supercomputing Research Center at ISC. So I'm extremely humbled and happy to be here. Today's lecture I'm going to be giving on is how and where memory is stored in the human brain. So, can you hear me?
Yeah.
Okay. In the back?
The neck maker. The neck maker is better.
Is this better?
Yeah. Make a light also.
So, my talk today is on how and where memory is stored in the human brain. And this is a subject that's very near and dear to my heart. I just spent the past 6 years of my life, almost a quarter of my life, in a lab at Duke University studying how memory resides in the human brain. But more so, memories are extremely critical for our daily function in life. We need memories to decide who we like. We need memories to decide who we hate. We need memories to decide who we love. We need memories to decide what type of decision we want. Do we want fruits or vegetables? So prior experience in the world is a powerful teacher in allowing us to make optimal decisions, to make optimal choices in the friends we make, the colleagues we trust. And additionally, memory is extremely important to who we are as human beings. So the memory I just shared about my father forms a very powerful moment in my life and bore the narrative in terms of why I decided to have an academic career. So memories are extremely critical to our personality and what makes us unique individuals. So just a primer on Duke University if you're unfamiliar. So Duke University is in North Carolina. It's quite well known for this chapel. I have to promote Duke University, given that I was there for 6 years. I obtained 2 degrees from there, and I still cherish it to this day. Just a couple of facts. It has 11 Nobel laureates affiliated with the university, 32 members of the U.S. National Academy of Sciences, 8 members of the U.S. National Academy of Engineering, And if you look at the world rankings, it's ranked in the top 30 based on any type of metric that you use. So it's a wonderful research university. So I know there's some high school students or master's students here. If you're considering Duke, I would wholeheartedly agree for that decision, and we can talk afterwards if you have any questions about enrollment or getting admission to Duke University. So where I completed my doctoral research was in Roberto Cabeza's lab. My doctoral advisor is extremely famous, primarily for his work on how the brain ages over time and how the brain's activity changes over time due to age-related deficits in cognition. The lab that I was in used functional magnetic resonance imaging, which I'll go over in a couple slides. But its interest was looking at how memory is stored in the brain, what happens when we remember things from our personal past, what happens when we forget things from our personal past, why we tend to remember certain things, such as powerful or unique events from our life, and why we tend to forget certain things that are mundane in our individual lives. And he's written several books, including Cognitive Neuroscience of Aging. He's written one of the premier handbooks, along with other authors, that serves as a textbook on cognitive neuroscience in the Journal of Cognitive Neuroscience, which is the premier cognitive neuroscience journal from MIT Press. He's been on the cover many times. The reason why I mentioned this is that the field of cognitive neuroscience is a relatively young field. It's only about 30 years old, and he made a profound impact on that field. So the questions of interest in terms of the lab, and I apologize it's cut off here, is basically threefold. There's a three-pronged strategy as to how the lab does its research. So the first question is how and where memory is stored in the brain. So it's a very fundamental question of where memories are stored in the brain, what goes wrong as we age, why certain memories are stored in certain brain regions and other memories are stored in other brain regions. Secondly, what are the external factors that influence what we remember and forget? Extremely critical. Why we remember certain things, why we forget certain things, knowing what influences that is extremely critical in understanding how the memory system works in the brain. And thirdly, any age-related deficits. So aging is a big component in the lab. He was one of the first to show that older adults, age 65 and higher, compared to younger adults, ages 18 to 42, they tend to recruit more brain regions. They tend to over-recruit in terms of their activity, these brain regions, to successfully perform as well, or if better, than younger adults. So they need more resources to do as well as younger adults. So he's called this the Herald model, hemispheric asymmetric reductions in older adults, and he's known for that. So there's some seats here for people looking. So before I begin at the main topic for today's discussion, I just wanted to give a brief primer on the anatomy of the human brain so that everyone's on the same page. So the human brain is composed of 5 lobes. Sometimes people consider the insular cortex as another lobe of the brain. You have the frontal lobe, which is here, and it's primarily responsible for executive functioning. So making a decision, having a plan in mind, carrying forth with that plan, high order reasoning, judgments and preferences based on what we like and what we don't like, primarily resides in this frontal lobe of the brain. Then you have the parietal lobe of the brain, which has been canonically linked to attention. So ability to pay attention. Your ability to pay attention to me right now, both spatially and visually, is highly dependent on activity in regions in this parietal lobe. Specifically, a region called the temporal parietal junction. So we have the occipital lobe, so our ability to process incoming information from our environment in a sensorial manner occurs through this occipital lobe. So vision occurs through the eyes, but then it's fed forward in a feed-forward manner to the occipital lobe, where the process of determining this is a laser pointer, determining the lines and orientations of this laser pointer is achieved in this occipital lobe. And finally, the temporal lobe is the region that I did my doctoral thesis on, and it's critically important for a couple of things. One critical important function it serves in the brain is for memory. So our ability to remember certain things, certain facts, certain events occurs in the hippocampus, which resides here in the temporal lobe. Our ability to understand conceptual knowledge, so knowing that this is a mic, knowing that this is a water bottle, so our real-world knowledge of the world occurs in the temporal pole, which is a part of the temporal lobe. So this is an extremely important lobe of the brain. This is an extremely important lobe in the brain that's important for memory and semantic knowledge of the world. And this will be today's discussion, right? This will be the predominant area with which I will discuss today. Apart from the major lobes of the brain, we have several other regions that are important. We have the cerebellum, which is located here in this cauliflower-shaped region of the brain. It's important for motor movement. So my ability to move, hand-eye coordination, is definitely gauged here in the cerebellum. The ability to have a basal ganglia, so is important given movement as well as hand-eye coordination and coordination with the cerebellum. So Parkinson's disorder, which you might have heard of, is largely a type of dementia. It's a progressive type of dementia, and it's a movement disorder that primarily affects the basal ganglia. The brain is an organ that's about 3 pounds, right? 3 pounds. But it holds as many neurons as there are stars in the galaxy. That's something to think about. If you take a moment and think about that, this 3-pound tissue is more complex than the most complex supercomputer that any computer scientist has developed. And I'm looking at my father right now because he's a computer scientist and he's disagreeing with me right here. But it is the most complex machinery in the world. So just a brief primer on the brain anatomy. So the idea is, what is cognitive neuroscience? So Dr. Ramarao gave me a good segue in saying that he might not have ever used the term cognitive neuroscience because it is a very specialized term. Part of the reason is that the field is only about 20 or 30 years old and was originally conceived by computer scientists, which will give some interest in the room. So computer scientists at CMU and Stanford, primarily Marvin, and at MIT, Marvin Minsky, Alan Newell, Herbert Simon, these individuals came together and they were all interested in the brain, and they came as more of a computer scientist and logician type of viewpoint. However, what they decided is that you couldn't treat the brain as a black box. You needed to understand the inner workings of the brain in order to determine how we are uniquely human. So they termed this field, cognitive neuroscientists, in the late 1970s, early 1980s. So what is this field about? So it takes real-world daily behaviors, such as deciding to pay attention. Maybe you've already decided not to pay attention to the stop, which is fine. Maybe you memory for a shopping list. So you're going to the grocery store, and instead of jotting it down, you're going to rely on your memory for a shopping list. So these are daily behaviors that I'm describing in the real world. Making a specific decision. So deciding whether to attend this talk or deciding whether to attend a colleague's birthday party, for example. Those simple decisions that we face on a daily basis that have minimal consequences are the goal of cognitive neuroscience. Another type of daily behavior, choosing an ice cream flavor. So choosing whether to taste ice cream or to consume ice cream, and choosing a particular type of flavor that you're interested in, very basic decisions, right? Very basic judgments. But this field is interested in how the brain arrives at those judgments. And finally, stopping at a traffic light. It always amazes me when I come back from the United States how there are no traffic rules here in India. So many people don't follow this daily behavior. But basically what this field wants to do is take all of these behaviors, right? And there are many more. There are millions of permutations and combinations of behaviors, right? Take these very simple behaviors that are extremely complex, right, in the brain, but are simple to us, and see how it resides in the brain. So what are the neural correlates? I.e., in other words, where do these decisions arrive in the brain? Where do they reside in the brain? What are the brain regions that are critical for making these decisions or making these executing these behaviors? And what are the brain regional interactions that allow us to execute these behaviors? And finally, how do they interact? And in turn, what is going on in the brain that allows us to make these specific decisions? And what damage could occur in the brain that will make us extremely faulty at making some of these decisions? So one could say that, so this area of the brain is a temporal lobe, right, which we just went over. Damage to this area of the brain may impair one specific behavior, in this case, memory for a shopping list. So if you probably all have heard of Alzheimer's disease. It's another type of dementia, the most common form of dementia, only 60 to 75% of the cases, and it targets a specific area of the temporal lobe, the entorhinal cortex. And we'll go over the anatomy in a bit. But this is an important region of the temporal lobe that is important for memory for our personal events. So imagine if you have damage to this region of the brain, you will have to rely on a shopping list to make your groceries, right, to get the groceries. So and then another question that's interesting, going this reverse direction, is do similar types of behaviors cluster together in the brain, right? So if we went in a data-driven approach, and we were a computer or a machine, right? And we wanted to know what types of behaviors cluster together or are more similar than one another, and what types of behaviors are dissimilar. We can use activity that corresponds to each of these behaviors to look at where they are located in the brain, and if they're located in spatially similar positions in the brain, then we could conclude in a more data-driven approach that these behaviors are similar. So if we go to this list, right, we could easily say, right, stopping at a traffic light and deciding to pay attention are two dissimilar behaviors. But they also have many commonalities. They both involve a decision, they both involve consequences, and seeing how these type of behaviors reside in the brain and what brain regions are critical is an important question. So that's a bit basic introduction as to what the field is primarily interested in. So we take real-world behaviors. How do we look at this in the brain? We look at it in a largely non-invasive manner. So I said these are very simple behaviors, such as stopping at a traffic light. But how do you mimic these behaviors in an experimentally controlled method or fashion to then study them in the brain? That becomes an important question. That's the important question. How do you do that? Well, there's certain tools that are existing currently, and I'll show you, we'll go through those tools. So this first one is event-related potential. So the brain, right, it's a three-pound tissue organ. It emits magnetic waves, correct? So me looking at you is a certain specific magnetic wave at a specific oscillation in frequency. And given that I know the specific oscillation of frequency in terms of spatial attention, I'll be able to conclude that the brain is undergoing some kind of process that it's executing for me to spatially orient to you. So event-related potentials take advantage of brain waves that are emitted to then look at the areas of the brain as plotted here in circles as to where they're located spatially. This type of technique has very good temporal resolution. So making these minute decisions or gazing to the left or right is in the order of milliseconds. So you need a methodology that is sensitive to these minute millisecond differences in attention. So this type of technique is very good. However, as you can conclude here from these example results, the spatial resolution is bad. So given that the brain is comprised of a myriad of regions, you want a method that has good spatial resolution to then conclude this particular region of the brain is important for this type of behavior. And that's where functional magnetic resonance imaging comes from. So you might have seen at Times of India that can herald a lot of news about showing these type of pictures and saying this area of the brain is important for this decision. It's a gross oversimplification of what's going on in the brain. However, I've been here since June 20th and I've seen already 6-7 articles in both of those periodicals. What does that say? A, that tells me that neuroscience is a very popular field, and B, that journalists don't always get it right. So what happens in fMRI is that it takes advantage of the brain emitting magnetic waves, but it goes a step further. So the brain is composed of water molecules. Water molecules, H2O. What happens is, as your brain is undergoing a particular process, such as the movement of this arm, oxygen comes from certain areas of the brain to those areas of the brain. It moves to those areas of the brains that are critical for hand movement. And by looking at the displacement of oxygen molecules in the brain using this fMRI machine, then you're able to show differences in activity. And that's what this is basically showing. What happened in a typical fMRI experiment is we stick people into this scanner, which is a 3 Tesla or even 7 Tesla magnet at some institutes, even in Holland. And we present them an experiment. So while they're in the scanner, they do a particular experiment. It could be a memory experiment, and I'll go over some examples of memory experiments. And then what we're able to show is activity as represented by these blobs on the brain, but they're basically subtractions that we're computing that look at differences in oxygen movement in the brain. This type of technique, as you can tell, has very good spatial resolution, but poor temporal resolution. And you can tell that here in that you can look at the specificity of this left area of the brain that you don't get here, but the temporal resolution here is on the order of seconds, not on the order of milliseconds. So that's the advantage and disadvantage. Finally, what is very interesting here is TMS, so transcranial magnetic stimulation. So what this technique is able to do is to use magnets to essentially null or silence a particular brain region. So what it does is it infuses a particular magnetic field, and that magnetic field disrupts neuronal activity in a particular brain region, and by doing so in this case here, in this right motor cortex strip here, by doing so you can silence it. And when you can silence that particular brain region, you can see whether it's critical for a particular function. So if I get rid of a particular brain region and the person that I'm doing this technique on cannot execute a particular type of movement or cognition, I can then conclude that this brain region is important for this particular function. So let's begin with a simple experiment. So cognitive neuroscience is very similar to physics in the sense that we have a very good intuition of how human beings behave. How do we get that intuition? From our own lives. Me understanding how people make decisions is very simple. So for example, I'm on a diet, or I should be, and I go to the ice cream shop, and I make a decision, okay, should I get ice cream, or should I choose a healthier option? What are the factors that go into the decision? It could be, is it my cheat day? So for example, have I been good on my diet for the past week, and this is the one day I want to cheat and take ice cream? Am I able to plan ahead and understand that if I do take the ice cream, I will feel bad afterwards? Right? These are the thought experiments we undergo as cognitive neuroscientists on a daily basis, to then design experiments to test where in the brain these type of behaviors reside. So I want to begin with a very simple thought experiment, and hopefully you will follow. So I want everyone to close their eyes, just for 10 seconds. Please close your eyes, everyone, just for 10 seconds. And as your eyes are closed, keep them closed, I want you to think of an event or an experience from your personal past, just for 10 seconds. Okay, open your eyes. Do we have any volunteers? What did you guys think of? Any memories? Any events?
Blank.
Blank. Someone said blank. Anybody else? Okay. In my class that I used to teach, I ran this thought experiment, and we came up with different answers. But this thought experiment allows researchers in the field to determine what type of memories people tend to retrieve, right, given a short period of time. And this 10 seconds is longer than what we give to participants while they're in that fMRI machine. We usually give them 4 seconds or 6 seconds. So these are the events you could have retrieved, right? A birthday party. So that's one thing I retrieved. I was at my nephew's birthday party yesterday. Or you could have retrieved a wedding day, if you're married. Or you could have retrieved a graduation ceremony. Or India winning the World Cup. Or taking an SSLC exam, which I think is about what is it, 8 months from now. Or a visit to Joe Falls, which you can't see here. But what is inherent to all of these events? They all seem different, right? But they're all very common in one or two couple of elements. So they all are memories that one could retrieve that are highly emotional, that are personally significant to you and to your individual lives. And all these events that you might have retrieved share three elements. There's a temporal aspect to it. So each event you might have retrieved has a particular time. They have a particular time stamp in your memory. So India winning, I think, was 4 years ago, 2 years ago, 4 years ago? 4 years ago, okay. Birthday party for me was yesterday. Wedding day, it hasn't happened yet. Graduation ceremony, for me, it was 6 weeks ago, right? So each one of these events that are retrieved have a different type of time stamp. The second thing it does, there's a social element to it. So each of these events that are retrieved, which are essentially memories, has a social component to it. Who was there? What did we talk about? What were the positions, the orientations of the individuals there? So this is the birthday girl, and then here are the positions of the individuals that she might have invited, right? And there's a spatial component. So what were the actors doing in that particular event? What was the spatial layout? So what I'm trying to drive home to you is that memories are composed of these three elements: time, space, humans. Those three components. And we didn't have to run an experiment that cost $500,000 or even more to run. We just did it here, right? We just did a thought experiment here. So I want to introduce a preliminary definition of memory, and it has three components. So it represents retrieval of a unique event from one's personal past. So there can only be one instantiation of that event from your personal past. So there's only one birthday party where that particular nephew or niece of yours turned 6 or turned 7. There's only one time in your personal past that you might have visited Joe Falls. There's only one time that in your personal experience that India might have won the World Cup and you witnessed. Every event is unique. And there's a reason why the brain treats every one of these events unique, and we'll get to that point in a few minutes. Secondly, they're often associated with a high degree of personal significance. So each of these events are emotional. You visit Joe Falls, you gained some pleasure, you went with some friends, it was a good trip. That's a highly emotional event. And thirdly, they're all comprised of details, as I mentioned, temporal, spatial, and social. So that was a long introduction, but I wanted to lay the groundwork as to why the study of memory is important and how we do that, how we study memory in the brain. How we study a concept is extremely abstract. And how we study that in the brain, and how we devise experiments in our laboratory to examine memories in the brain. So I first for today, want to look at neuropsychological evidence of brain structures that are important for memory. So the recent 30 years have witnessed an expansion as to the role of technology in the study of memory. But this work here was done in the 1950s from patients that had lesions to particular areas of the brain that were found to be important for memory. Two such patients, if you don't take home anything from today's lecture, you should say that patient HM, which we'll go over, is the most important contribution to our understanding of memory in the past 50 to 60 years. Secondly, what I want to do is, oftentimes, when I say, okay, I got my doctorate in cognitive neuroscience, I study memory, they'll say, how do you study memory in the brain? So what I want to do is lay out some methods by which we analyze how the brain treats events that we remember versus forgotten. The idea of certain types of memories that are associated with rich contextual detail versus those that are devoid of contextual detail. And true versus false memories. So memories that we think we experienced in the past, but we really didn't. And lastly, given that this college has a heavy engineering and computer science background, I want to look at the recent interactions between computer science and cognitive neurosciences, specifically two techniques, one looking at multivariate pattern analysis of neural data, and more excitingly, in the past year or so, the use of graph theoretical measures to examine how the brain's network is structured. So, Lesion-based studies are one way to show structure-function relationships. So what happens is that researchers, they recruit patients, right? They recruit patients with lesions that are focal to specific brain regions. In this case, we're looking at the parietal cortex here that we mentioned in green, which I know is difficult to see in the back. So these set of 5 patients had lesions to this particular area of the brain. Then what is taken is great care is taken to ensure that there's maximal overlap in the lesions in the 5 patients or so that are brought into the lab. And what they provide the patients to do is a simple test. So in this case, it was a spatial attention task divided into 5 components. There's a warning cue, then a delay, then a cue for their attention to go this way, to their left, and then to make a decision whether the stimulus in this screen was on the left or right. So basically what they're trying to do is, do these patients that have a lesion to the parietal cortex, do they still retain the ability to attend either to the left or either attend to the right? So that's the power of lesion patients, right? These studies are the gold mark of, or the gold standard rather, of neuroscience, because you can look at one specific region's contribution to a specific type of cognition. And that's what we happened in the case of HM. Henry Malazon, was an American citizen. It's a really tragic story, but he experienced a bicycle accident around the age of 7. And as soon as he hit after the accident, he started to experience seizures. And he started to experience so many seizures due to that accident that he told his mother, I need to go to the hospital. So he goes to the hospital, and they say, okay, Henry, you have epilepsy. And epilepsy is a disorder which is characterized by frequent seizures, right? And he went to a specific physician, Elizabeth Scoville, and what she did is she removed the medial temporal lobe of his brain. And the seizures stopped. So all's good, right? Well, not really. So what happened is, what she did is, so this is the brain of HM, and this is a normal brain. You see the normal brain, everything's intact, all the regions of the brain are intact. But in HM, in order to solve the issue of having multiple seizures, the physician removed this part of the brain, the medial temporal lobe. The medial temporal lobe containing regions of the hippocampus and entorhinal cortex. She removed that completely. Seizure stopped. What's interesting is that follow-up studies with HM on tests of intelligence and tests of memory in the late 1950s showed a problem. So in this table, you see that this red box here is highlighted HM, and I know it's very difficult to read, but he was okay on the intelligence test. So IQ test, fine. He's okay. Memory test, he failed. So he's about 2 to 3 standard deviations below the mean of a normal control. So this is the first evidence showing that if you remove this part of the brain, if you just completely remove it, your memory is going to impair. This is the first evidence showing that medial temporal lobes are important for memory. There was another patient, KC, this time a Canadian citizen. His name was Ken Cochran. He also experienced another motorcycle accident. He had traumatic brain injury. Soon after, he experienced epilepsy and similar type of case of seizures. So he goes to the doctor again, and they removed his, in this case it's a radiological convention, they removed the left medial temporal lobe. Similar type of procedure. So based on HM, based on the first patient, we would predict that his memory would be shot, right? It would be impaired. One thing that was interesting is that it showed subsequent tests in KC showed that memory is not unitary. That he was good on some types of memory, but not all types of memory. So similar to HM, his episodic memory, or his memory for his personal past, these unique events, was completely gone. And I'm sorry, you can't see that here. Well, you can here. So this is episodic memory. This is completely gone. But this is another type of memory here, which was completely spared. And the type of memory that was completely spared is what we call semantic memory. So this is memory for knowing that this is a microphone, or knowing that this is a water bottle. This is not a unique event. There's no timestamp corresponding to your memory of what this is or what this is, right? That's the big differentiator between semantic memory and episodic memory. So semantic memory has no timestamp. There's no social component. There's no spatial component, but episodic memory has those three components. So that's what this patient showed. It showed that not all memories reside in the same place of the brain, but rather rely on different places of the brain based on the demands they impose on the brain itself. So this is the point I just made here. So similar to patient HM, patient KC exhibits deficits in episodic memory, but not semantic memory. And I hope that's clear, that distinction. It's a very important distinction that we learned from patient KC. So these two patient studies show that there are multiple memory systems in the brain. So if you think about it, you have one long-term memory structure, right? Knowing that you still rely on this, knowing that this is a bottle of water, or this is a laser pointer, relies on long-term memory. But then you can further subdivide long-term memory into declarative memory, or memory that you can talk about in a cogent manner, and memory that you cannot talk about. So for example, semantic memory and episodic memory are two forms of declarative memory. You can describe what this is, you can describe a personal event such as a visit to Joe Falls, but there are different types of memories that you cannot describe, not verbally. So if I ask you, how do you ride a bike? Can someone tell me how to do that? It's very complicated. How do I ride a bike? Okay, you take one pedal, then another. That's not a good definition. It's not a good definition because it relies on a memory system that is based on classical conditioning and priming. It's a very physical type of memory that has no timestamp, that has no spatial component, has no social component. It is procedural memory in this case. So there's a procedure that you master, but then you can't really talk about. So that was what's important in terms from these two patients in showing that there are two different types of memory in terms of long-term memory. Okay. So that was a brief background in terms of what patients have showed us about memory. So now the interesting part comes from there's no patient HM and KC. Of all the studies show with lesion patients, there are only about 5 or 6 lesion patients. Why? Because damage that is so localized to a particular area of the brain, in this case the medial temporal lobe, which is so inside of your brain, is very difficult to find. You won't get patients like that. Very rare. So devising experiments in healthy adults, such as ourselves, is extremely important to furthering our understanding of how memory resides in the brain. So how do we do that? So before I go into that, I want to introduce a diagram of how our experiences in the world are transformed into memories. So suppose you've experienced this event, right? Coming to a lecture, hearing me talk. Then you go to your dorm, or you go to your home later this evening, and you try to remember, okay, what was said, what was said about memory, was I thinking about the dinner instead of listening to the talk, things like that, right? So let's talk about this pathway. So let's go back to our example of a birthday party, okay? In my case, my nephew's birthday party yesterday. There are various details of this event that while I was experiencing it, was encoded in my brain, that I was encoding. I was taking all the sensory knowledge and trying to encode it in my brain. So one part is the spatial details of the party layout. Okay, these girls were here, these girls on the right of me, I was in the center, there was a cake in front of me, my mother was to my left, my father or grandfather was to my back right. All the spatial details are encoded in the upper portions of the brain, the dorsal. There's a dorsal pathway of the brain that's important for spatial layout. Then you have elements of the party layout themselves. Okay, what elements were there? There was a cake, there was a balloon, there was some streamers, she was wearing this type of dress, there was a candle. So all the objects that are associated with the concept of birthday party get stored in the ventral pathway of the brain. So you have one pathway of the brain that's important for interrogating the Fourier relationships and the spatial relationships of an event, and you have a ventral pathway that's important for understanding the object and the specific event itself. Then you need one part of the brain that beautifies all this information. So it's not enough if you have spatial information in one part of the brain, object information or elemental information in another part of the brain. You need one region of the brain that bounds these two types of units together to form a coherent representation. So what is that? That's going to be here in the medial temporal lobe. So what it does, it forms a schema or a spatial layout or an idea, a concept of a birthday, and then what it does is it goes to birthday party, and then it embeds that event representation with all these events that are, with all these information specific to that event. So it takes these two event pathways and forms a bound, unique, coherent representation of this memory. And what do we call this process? It's called episodic encoding. See, the idea of incorporating information from our environment and how it gets rested and laid down in the brain is called encoding of information. So then over time, what we get is memory consolidation. So over a week or 2 weeks, you don't think about it, but the information that you've already encoded becomes stronger. It becomes stronger through this consolidation process. So the way you can pictorially represent that is by imagining these arrows as bigger, right? These trace elements of the memory as bigger. So the memory trace becomes stronger and stronger over time. Which is why you might ask, why our older adult loved ones tend to remember events that are more distant in the past than more recent in the past. Finally, we have to retrieve, right? So if you're at your house tonight and trying to remember what was said during this talk, you need cues. So suppose you're in an environment and it's been about 6 months since you've been to this birthday party, but then on the street you see this candle. You see this candle, and what it does, it serves as a cue. And that cue serves to tag the memory of the birthday party that you already experienced. And what it does, it reactivates all of these elements. And this happens very often. You know, I'm driving with my father through Bangalore, and he was born and raised here. And each time we pass the Downsauda, a memory pops into his mind. Why is that? The Downsauda serves as a cue. Maybe he had a good experience there, maybe he had a bad experience there. But there are all these cues in the environment that serve to help us to retrieve and subsequently reactivate memories that we've already experienced. So how do you do this in the scanner? So as we mentioned, we stick participants in the scanner, right, in the fMRI machine. How do you do that when they're sitting still? How do you mimic information from the environment? How do you mimic my father's ability to use the Downsauda as a cue to remember some things in the past? How do you do that? Well, this is how you do that. This is what has been done. Very simple. There's a term in the literature called ecological validity. And that basically is saying, are you conducting an experiment that is replicable, that really replicates how we tend to experience our natural world? And the basis of these experiments is that they tend to be highly ecologically valid. So what they tend to do is present lists of words. So mustard, giraffe, moon. And they ask you to try to either memorize those words, try to process those words in a particular manner. For example, is this a noun or adjective or word, verb, part of speech? So semantic processing of those words, or perceptual process. Is a mustard seed bigger or smaller than a shoebox? Just some way of orienting you to process that word. Then what they do is, you're in the scanner, right? So they collect neural responses to each of these events. So I present you mustard, and you imagine it being bigger or smaller than a shoebox. And while you're doing that, I'm collecting the brain data. I'm collecting data from the brain activity to be specific. And then during retrieval, I test your memory for those events. And what I do is I represent all these lists of words, and I introduce new words. So I might introduce here temple. You didn't see that in the original list, so it's a new word. So during this memory test, what you're asked to do is tell me whether This is a word that you saw before or a word that you've not seen before. And then what you could do is then reclassify these visual events as either remembered or forgotten. Is that clear? Remembered or forgotten. Okay, so then based on this memory test, I could say whether you remember this word, or you forgot this word, or you remember this word, or you forgot this word. And then what I can do is I can contrast brain activity that I measured for items that you remembered versus items that you forgot. And this is a subtraction logic that we employ to look at areas of the brain that are critical for memory success and memory failure. And that's what's done here. So you can see these areas of the brain, this area of the brain in the left prefrontal cortex, exhibits higher activity for remembered versus forgotten. That's basically looking at the subtraction of the activity for those remembered versus forgotten. And you can do the same thing in the left medial temporal lobe. So you can begin to get an idea of the types of brain regions that are critical for memory success. But there are different types of memory quality. So when I asked you to think, there could have been some memories that you really remember. You really relived that event. And some memories that were extremely vague, that you really couldn't fill in the details. Well, this describes two types of memory. So not all memories from your personal past are equal. Some are stronger than others, some are weaker than others. You might have experienced this before. So one could be recollection, one we term recollection. So that is the retrieval of an event from your personal past where you can remember all the contextual details. The people that were there, where you were, what you were doing, the time of day. All these rich contextual details that make an event in your personal past very rich and produce the feeling that you've re-experienced an event. The other type of event could be familiarity, so weaker form of memory. So here, I might not remember a birthday party. And there could be several reasons why I might not remember the birthday party. Maybe I've been to many birthday parties, and as a result, there's interference due to the shared elements across those birthday parties that I've experienced in the personal past. So how do you get this in the brain? How do you determine between true memories or memories that are really experienced versus those that are partly experienced? So that's a step beyond just saying remembered versus forgotten. That goes beyond and saying, does the person really remember that particular event? So this is a study by Charan Ranganath and colleagues in 2004. So what he did is he presented words on the screen for a brief period of time and asked them to do one of two tasks. In the green, he asked them to determine whether a nickel, the word that was presented, is larger or smaller than a shoebox, and words that are presented in red, are they living or non-living? So two types of encoding judgments. And then he simply asked them, rate the confidence with which you remembered that item during a memory test. The idea being that if you really recollected that experience in your personal past, you would have a higher confidence associated with that event than if you only familiarize yourself with an event, you would have a lower confidence. So what Ranganath and colleagues were getting at here is the idea of, can we access the brain regions that are important for those memories that we really experience? So what you can see here is that plotted here is new and old. So they basically represented these words and asked them to say whether it's an old word or new word. And as you can see here, definitely old, which is, I definitely remember seeing this word, right? Is characterized by a higher proportion of responses that are correct, right? So this is an index of a recollection type of memory process versus a familiarity type of memory process. So what you can do here is then locate the regions of the brain that are important for familiarity versus recollection, and they're both. Then you could look at dissociations between those two types of memories. So what this showed us is that there are two types of memories, and even in the medial temporal lobe, there's dual process models of memory. So if I gave you this, if you were shown this fruit basket here, this is from your visual input, it goes to two different pathways in the medial temporal lobe, which then forms your memory. This area of the brain, the medial temporal lobe, the parietal cortex, encodes the color or the featural properties of those events, whereas the fruit basket layout is encoded or processed by a different region of the medial temporal lobe. So this is the parietal cortex in blue, perihippocampal cortex in yellow, and they both converge to form a unique memory representation in the hippocampus. So I want to conclude this section with one other interesting study looking at true versus false memories in the brain. So as I mentioned before, there's some memories where you think you remembered an event from your personal past, but you really didn't. And this is a study from my doctoral advisor's lab, where he presented during the encoding of information, during the encoding of memories, lists of words, so horse, chicken, sheep, goat, and then he tested their memory for these specific words and asked them to say whether they were sure they saw a particular word or that they were unsure. So chicken, yeah, they should have said it's a true memory. Tree, it's a new memory. Or cow, it's a false memory. The idea is that if you're tested on words that are related to the list of words here, so cow in this case, and you say you're old, you're pretty sure that you saw it before, that's indeed a false memory. It's related to an event that you experienced before, but you really never experienced before. So this is an exciting area of research because then you can look at true recognition versus false recognition and look at the differential neural correlates behind those two types of memories. So I want to share one other thought experiment which drives home the point of why it's important to study true versus false memories. So suppose you park your car at this parking spot. It's a very common occurrence. And suppose you park this at day 4, right? And then you come back on day 4, and then you have trouble finding a parking spot. There are 4 levels to this parking deck, but you don't know which one where you parked. Why is that the case? You have a memory that you parked in this particular parking spot, but there's interference from memories from other days where you parked in similar but not identical parking spots. So what is going on here? Well, there's interference, and this occurs on a daily basis. So if I told you 3 days ago, what did you have for breakfast? Could you tell me? No, right? No. Why is that the case? Every day you have breakfast, right? If you ask me today what I had for breakfast, I don't know. I have a horrible memory, which is why I study memory. But
because you eat.
Yeah, I didn't eat. Yeah, that's what I'm saying. So the idea is that if you experience an event multiple times in your life, there's going to be interference that's produced from experiencing those multiple events, and that your interference is not good and for your memory. So how does the brain overcome this? It does what's very interesting. It does something called pattern separation. So the 4 parking lots in the previous slide are all very similar. They all have similar color, similar cars. They have a roof. So they're all very perceptually and conceptually similar. So we need the brain to be able to tease apart these memory representations in order for us to lead to the proper decision. So suppose you have A prime, A double prime, A triple prime, corresponding to the 4 parking lots. What the brain does is it takes this input and tries to minimize the overlap, and in turn maximize the differentiation between those brain representations. And by doing so, it leads to better and better memory when you're deciding between events that are really similar, but deciding which one you should go for, or which one you experienced before. And this is interesting when you look at, this is tested by looking at 2 different ducks, or 2 different light switches, or 2 different apples that have different perceptual orientations. So I want to conclude today with something that is very interesting, but very recent, and it looks at interactions between computer science and cognitive neuroscience. So one thing that's big these days is machine learning. You can't go anywhere without the idea of artificial intelligence in machine learning. You can look at machine learning as a neuroscience, as an advantage for the study of the brain. Why is that? The brain data that we get is in the order of sometimes terabytes. There's so much data. We're collecting data from 136,000 voxels in the brain, and each voxel contains that much more information. So the idea is that if we want to see how the brain is organized, what we can do is train a machine, a support vector machine, or a classifier on a set of particular brain data, and then test that data to see whether the activity or the patterns of activity that we collect from humans correspond to item A or item B. This is clarified by this example. So a big problem in neuroscience is whether there are different parts of the brain that correspond to different types of stimulus classes. So is there a separate part of the brain for the processing of faces? Is there a separate part of the brain for the processing of houses? And is there a separate part of the brain for processing of scenes? Right? Does the brain process all three types of stimulus classes differently or similarly? And machine learning can help answer that question. So suppose I give you two types of categories, a bottle and a shoe, and I give you a series of bottles and a series of shoes, right? And each time you see a bottle or a shoe, I collect brain data. What I can do is then look at brain activity for different real-world categories by using this machine learning algorithm. And what I first want to do is collect all the brain data, all the brain activity from each of these categories, and then sort them in a vector, right? From voxels V1 to Vn, and then use this as a training set to then test the machine classifier to see that if I give you a random set of voxels and neural activity, can you tell me whether the human subject saw a bottle or a shoe? Right? So each bottle has a separate type of activity as outlined by this 6x6 matrix here of voxels, and the shoes have different types of patterns of activity. So basically what it does is you can test a particular type of vector and see whether it's either a bottle or shoe without you determining whether they actually indeed saw a bottle or shoe. So this is important when determining whether in memory, whether people are saying indeed they're remembering or they're just simply lying. So a big issue is we ask the participants, did you see this or not? It could be lying, right? But using machine learning, we'll be able to assess those patterns of activity corresponding to when they're really remembering and when they're really forgetting, and then test that activity on a different type of frames. So this is one example of the interaction between computer science and cognitive neuroscience. Another example is networks. This is a very clear example and a very straightforward example. So what we tend to do is parcellate the brain into these different anatomical regions and then use the histological and imaging data to construct a network. You construct a network of brain regions that are connected either by their structural means or by their functional connectivity means, and then you can construct a network to look at to derive graph theoretic measures to look at whether there are modules in the brain, shortest path algorithms, how the brain is efficiently optimizing information transfer flow. So this is another type of example. And what's going increasingly, the idea is that you can use a lot of these graph theoretic measures. So here in graph A, what you see is that there you have several nodes here, and each node is connected to another node by an edge. And what you can do here, just using the structure, is to calculate a whole host of graph theoretic measures looking at modularity, so the extent to which a network is modular or has these modules or the number of modules, how each module is connected to one another, whether there's a hub within a particular module, and whether that hub might be low degree or high degree. So this is extremely important when you want to differentiate between two types of patient populations, either a diseased patient population or a healthy population, to see whether they differ in this type of network property. So I want to close on this slide by showing that in the brain, what this is showing how the brain is a network of 5 different modules that are connected to each other, but that also follow the anatomy of the natural brain. So graph theory is important for confirming that different regions that we will always assume to be connected with one another are indeed connected with one another. So I'll close with that. And just some broad conclusions. One thing I want you to take away is just 3 take-home points. Memory is not a monolithic phenomenon. You can indeed study it in the brain using these types of experimental methods. The hippocampus is extremely important for memory. whereas the rhinal cortices are more for familiarity-based memory. And there are indeed new tools grounded in computer science, which I think suit the talents and the strengths of the Indian subcontinent in terms of making fundamental contributions to cognitive neuroscience and to also how memory is stored in the brain. So with that, thank you. Thank you very much.
Good evening, ladies and gentlemen. We had a great opportunity to listen to a wonderful talk on how memory is stored in the human brain. It has been very informative and memorable talk, sir. On behalf of Bangalore Science Forum and all of us here, I propose vote of thanks to Vijeth Iyengar. Tomorrow's talk by Dr. E. Padmanabhan, former Director, Indian Institute of Science, will be speaking on translational effort in health research. All are cordially invited. Any questions, please?
Thank you, Vijeth, for thanks a lot for a wonderful talk. I have one question. How the memory is coded in the brain? Is it chemical or electrical activity or something like that? How it is coded?
Yeah, that's a good question. So the question was something that I neglected in the talk. I'll stand up so everyone can see. It's how are memories chemically stored in the brain? Well, they heavily rely on neurotransmitters. So one particular neurotransmitter is acetylcholine A, which is a neurotransmitter which helps these memories to get laid down or these traces to form naturally as a product of experiencing information from the environment. At the level at which my thesis was on was more systems level instead of molecular, but acetylcholine A, the use of neurotransmitters is important when looking at when examining how traces are formed in the brain.
What is brainwashing?
I don't know.
You know better, you had brainwashed.
All brains are brainwashed.
You are from Neuroscience, that's why I am asking this question, sir. Is brain transplant is possible in future, like other organs?
That's a really interesting question. There's a faculty member at Duke, whose name is Miguel Nicolelis, who's been able to control robots with mouse movements, with brain movements of mice. I don't know about a full brain transplant. I wouldn't want to have a full brain transplant. I think that technology or that even the use of that is, that's unfathomable to me. I wouldn't want to have it because then you lose the unique sense of self. I think with that.
If it is so successful in that, so what happens to the memory, from transplanting one memory, one brain to another person? Is that memory is going to keep, remains in that brain only when you transplant?
That's an interesting question. I think what your question is trying to get at is how synchronized are each of our individual brains? And there's some research from Uri Hassan's lab at Princeton University. He did a very clever experiment, which was in Science magazine. He showed the good, bad, and ugly movie to subjects, such as this, and then he measured brain activity during certain portions of the movie. Suppose it was a sad clip or a happy clip or even just a mundane clip, right, boring clip. And then what he did is he looked at the patterns of similarity, brain similarity across the individuals, and then using a method called inter-subject synchronization. And he was able to find that there's a great degree of synchronization. Whether that applied that, but that's only for human perception, right? Me seeing you seeing me is going to be a very similar type of product in our brains. Memories are extremely complicated, and I'm trying to get that through this talk. in that it's not just one dimension that memories operate under, it's multiple dimensions, and each person experiences the constituents of a memory in a different manner. So for me, some things might be extremely emotional. For you, they might not be emotional at all. So my hypothesis would be, in your case, if I were to transplant a memory from me to you, it wouldn't be treated the same. That's my hypothesis.
But wait a minute, anyhow, what is called the Madhyatman has given the answer. First transplantation was on Ganesha, I know. They got it. And that was much better. That's why I always say the first one, we do not know exactly.
Scientific.
You were talking about neural imaging, where we can observe the areas, but can you also use optogenetics by switching on single neurons to see what is the effect in your graphical circuits and neural network circuits? Is it possible in the memory area?
Yeah, no, it is possible. There's a group at MIT that, so I'm not familiar with optogenetics in terms of at a very granular level, but there was a group at MIT that was able to switch on and off in memory through optogenetics. So yes, that is possible. That has been done before.
Sir, I have a question on memory recollection. So, how is the memory recollected in a visual manner? I mean, I sometimes, as you said, I might recollect a birthday party in a visual manner. Where is it displayed, or how is it displayed through our memories?
So the question was, is how memories instantiated in the brain, or visually? Was that the question?
Yeah.
What happens is, when you are retrieving a memory, so if I cue you with the word birthday party, and I ask you to relive that event, it reactivates those components that were originally encoded while you originally experienced the event. So the particular type of individuals that were in the event, the spatial layout, all of those traces that you originally laid down are then reactivated based on that cue, right? And it's the collection of these reactivated traces that leads to a recollection type of feeling.
Oh, it's just a feeling.
That's a big debate. So there are two debates. One is, a lot of these reports are subjective, right? And there's another debate is whether you need consciousness in order to remember. In terms of the first point, you have these new tools, machine learning, and even looking at representational similarity analysis in terms of neural profiles that give you objective measures of whether they're indeed remembering or not. So that's where computer science techniques can come in handy. On the second thing, my doctoral advisor in a paper in Journal Neuroscience showed that you don't need consciousness to be able to remember. But that's another big debate we can talk offline.
Thank you, sir.
So while coming to the point that we see an object and it's separated as a spatial and it recognizes with a different color and the recognition, is there a special method to store the memory? I mean, in the sense in the brain, how does it store the memory and separates out the color and the distribution of the object?
Yeah, so there are two pathways in the brain. There's the dorsal pathway that takes care of the spatial properties of an event or an object. So knowing the spatial properties of this, that it can lay flat on a flat table and that it's curved, that's the dorsal pathway takes care of those spatial properties. Then you also have the ventral pathway, which takes care of the elemental properties, that this is blue and that this has a kingly, right? This has some sort of plastic wrapper. Those two converge at the hippocampus, right? So what your brain is doing is it's collecting bits of information from an object that you've seen or that you're seeing, and then what it does is it provides a bound representation of those informational bits. And that's the job of the hippocampus. It binds information both from space and time.
So is there different neurotransmitters for each of it to store, or is it the same transmitter?
I think there's I I can't say this definitively. I think there's a common substrate by which memories are stored using a common. But again, I can't speak to that. My dissertation was more on the systems level than molecular, but that's a whole host of issues.
You started at the beginning by talking about two things. One is...
Can you speak up?
I'm sorry. You start off by saying that the articles that you read in the Deccan Herald and the Times of India were largely wrong, if I may simplify. Is there any article or book that is useful, even if it's not completely up to date, that gives a reasonable thing of representation of both neuroscience and cognitive neuroscience? And I have a second question. You also, at the beginning of the talk, you said you're going to talk a little more about dementia, Alzheimer's and other forms of dementia. If you can expand on that a little bit.
Yeah, the first point, you still have time for a PhD. In terms of what I meant by that is, and there's brushback now. I think there's a desire to have the community obtain a very simplistic understanding of how the brain works, right? And that's the problem. That's one of the problems with fMRI. You'll see an activity blob on the brain, then it's tied to memory. Then you say this is a memory region, or this is an emotion region, or this is an attention region, or this is a decision-making region. That's incorrect. What I was trying to get at there is, the brain is a highly interconnected system, right? For memory, you need the occipital lobe. You need regions that code information about an event. You need regions that help to bind these information from disparate pieces. So, for example, if you hear a bell, right? If you hear a bell, then the auditory associations of that bell are laid down in the auditory cortex, right? Not the hippocampus. But those two regions interact when you're trying to retrieve a memory. So, that's the answer to your first question. There's a gross simplification of fMRI results, but the field is moving away from that. And journalists are increasingly taking stock in trying to tell the real story of what the particular paper was in. In terms of your, so if there's a reference, I could recommend the textbook that my doctoral advisor was a co-author on. Has a 20% market share.
Businessman.
But it's a really good book that grounds you in the fundamentals. In terms of Alzheimer's dementia, what I wanted to get at, I didn't have time. What I wanted to get at is to see how fMRI evidence has shown that there are specific regions of the brain that are critically first affected by Alzheimer's that then have downstream cortical impacts from there.
Which are those?
That have downstream impacts.
Which are those?
So the particular region I mentioned, the entorhinal cortex, which then feeds information to the hippocampus. So if the region of the brain that feeds information to the hippocampus to store memories is affected, then memories themselves are going to be affected. So that's what I was trying to get at.
Thank you very much for the wonderful lecture. I've got two small questions. Is the brain completely mapped for various activities? One thing. Second thing is about, we are always talking about the memory. Where is the processor?
That's an interesting question. To answer your first question, no. This field of using fMRI is only 20 years old. 20 years old. It's younger than I am, right? So the brain isn't mapped in terms of its action and the processes that it undertakes. In terms of the processing, I didn't go into this, but there are two types of maps that you can obtain from a brain. There's a structural map and then there's a functional map. So suppose Bangalore, Hyderabad, Banaji, whatever, right? Those are the cities, those are the brain regions. The highways that connect them are in the brain are called white matter tracts. And these are the processing units. These are the units, these are the structures rather, that allow information transfer to go from one region to the other. So that's your processing unit. That's the software, so to speak, right?
Sir, generally the memory level changes from one person to another person. What is the factor behind that?
Say that again.
Memory level changes from one person to another person.
Memories.
So, level of memory.
Oh, okay.
What's the factor behind that?
So there are several factors. One could be, you might remember something because of an extremely bizarre, unique event. right? So how many times am I going to come here and give a lecture here, unless Professor Rama deems me worthy of it, right? So this is a very unique event for me, so I'll remember it very richly, right? These interactions, these engagements. The other part could be it's highly emotional, personally significant, extremely unique or bizarre, and these help enrich our memories and how they're stored in the brain. Compared to brushing your teeth, eating breakfast, these are things you do on a daily basis. There's no attachment, there's no significance to those activities, so your memory for those activities are going to be very poor. So those are some of the external factors.
Okay, I think, no, wait a minute, there's no problem.